On the left, there's the female gamete, the secondary oocyte, and it is massive. We're talking roughly 2,000 times greater in volume than the sperm. Why? Because the oocyte has to do the heavy lifting, providing all the cellular organelles, the nourishment, and the genetic programming to support an embryo for nearly a week. Then, on the right, you've got the sperm, tiny, stripped of all excess baggage, highly streamlined and driven by that whip-like flagellum. Its one and only job is to deliver the paternal chromosomes. And talk about a brutal numbers game. For that tiny sperm, this is the ultimate microscopic gauntlet. Out of the nearly 200 million sperm introduced in a typical ejaculation, only about 10,000 even make it into the uterine tube. And from there, fewer than 100 actually survive the trek to reach the isthmus where the egg is waiting. To put that into perspective, if a male's sperm count drops below 20 million per milliliter, he's often considered functionally sterile, simply because there just aren't enough cells to survive the journey to the finish line. So how do those few surviving sperm actually pull it off? Well, it all comes down to a really groundbreaking concept called the Glycan-First Model of fertilization. Before a sperm can even think about binding to an egg, it has to survive the hostile environment of the female reproductive tract, and it pulls this off using sialic acids. These are 9-carbon monosaccharides that sit at the very tips of glycan chains, creating this thick glycocalyx coat right around the sperm. This glycan coat is literally a biological cloaking device, and it's the absolute star of our story today. Let's walk through exactly how this protective coat works in 3 stages. First, the sperm gets coated with these sialic acids while it's still maturing in the male reproductive tract. Second, as it enters the female tract, this sialic acid cloak interacts with specific immune receptors. They're called SLex. This basically broadcasts to the female immune system, hey, don't attack me, I belong here. It suppresses the immune response so the sperm isn't just instantly destroyed by white blood cells. And finally, stage 3. To actually fertilize the egg, this cloak has to be dynamically stripped away in a perfectly timed process called capacitation. It is a stunning sequence. The dual threats, oxidative stress and inflammation. But what happens when that sequence breaks down? We got to talk about the villains here. Infertility is a massive global health issue, affecting roughly 15% of couples of reproductive age, and in up to half of those cases, male factors are the primary contributor. Now, even with all our advanced tech, a lot of these cases are labeled idiopathic, meaning unexplained. But modern science is finally showing us that the explanation usually lies in invisible cellular-level damage. A huge culprit in all this is oxidative stress. This happens when the production of reactive oxygen species, or ROS, completely overwhelms the body's natural antioxidant defenses. And this is heavily influenced by exogenous modulators, basically real-world external factors. We're talking cigarette smoking, heavy alcohol consumption, exposure to environmental pollutants, poor diet, obesity. All of these trigger the overproduction of those harmful ROS molecules, which ruthlessly attack the fragile sperm. And it gets worse, because oxidative stress and inflammation actually act together in this vicious cycle. The ROS triggers the release of pro-inflammatory cytokines like IL-6 and TNF-alpha. These cytokines then sound the alarm, recruiting more leukocytes, white blood cells, into the reproductive tract. And what do activated leukocytes do? They generate even more ROS to fight off what they think is an infection. This self-sustaining loop destroys the sperm's delicate lipid membranes, literally melts away that protective glycan cloaking device we just talked about, and fragments its DNA. Measuring the damage, biomarkers in action. So, the crucial point is, if this damage is happening down at the chemical level, how do clinicians actually see it? Well, they use specific biomarkers to measure the exact type of damage that's occurring. For example, MDA is a byproduct of lipid peroxidation. It tells us right away if the sperm's cell membrane is being oxidized. Then you have 8-OHDG, which is a direct marker of shattered fragmented sperm DNA. And ORP, or oxidation reduction potential, is a global marker. If the ORP jumps above 1.34 millivolts per million sperm, doctors know there's a severe global oxidative imbalance that is actively destroying the sperm's functionality. But diagnosing this isn't just about checking one single box. As modern research points out, measuring just an oxidative marker or just an inflammatory marker on its own usually leads to underestimating the real pathogenic burden, because these forces feed into each other. Doctors absolutely have to look at the combined oxidative inflammatory profile to truly understand why a patient's sperm are failing to reach and fertilize the oocyte. The final battle breaching the zona pellucida. Okay, let's return to the heroes of our journey, those very few sperm that managed to evade the immune system and finally arrive at the egg. Now they face breaching the zona pellucida, which is this thick, glycoprotein-rich envelope protecting the oocyte. And this is where the Glycan-First model reaches its dramatic conclusion. Remember that sialic acid cloaking device that kept the sperm alive earlier? Well, now it's actually in the way. So during capacitation, the sperm has to shed this protective mask. This unmasking allows for the acrosome reaction, where the sperm binds to the egg's receptors, literally ruptures its own head, and releases powerful protein-digesting enzymes to melt a path right through that thick envelope. But here's a truly wild fact. This is not a solo victory. The egg is surrounded by a dense layer of follicle cells called the corona radiata, and a single sperm simply doesn't carry enough enzymes to break through that on its own. Dozens of sperm have to release their enzymes, specifically hyaluronidase, essentially sacrificing themselves to digest the bonds between those follicle cells. They clear the path together so that just one single, intact sperm can finally make it to the oocyte membrane. It's an incredible team effort. Let's see how this builds, because the moment that lucky sperm makes contact, it triggers a rapid, unstoppable cascade. Within seconds, the acrosome reaction releases those enzymes. Over the next few minutes, the sperm penetrates the zona pellucida and its membrane actually fuses with the egg. This fusion is the literal spark of life. It instantly activates the oocyte, causing sodium ions to rush in, depolarizing the membrane, which then unleashes a massive wave of calcium. Over the next few hours, this chemical reaction permanently hardens the egg's outer shell, completely locking out all other sperm. The future of fertility, precision medicine. And this brilliantly illustrates why understanding all this intricate cellular biology isn't just some academic exercise. It shows us how biomarker-driven therapies can intercept and fix that vicious cycle we covered earlier. If a patient's ORP or MDA markers are elevated, we don't just guess anymore. We can deploy targeted antioxidant therapies like vitamin C, E, or QQ10 to neutralize those free radicals. We can use anti-inflammatory interventions, shifts toward a Mediterranean diet, or even surgical varicose cell repair to eliminate the source of the oxidative stress. By restoring the biochemical balance, we can actually restore the sperm's glycan armor and its ability to fertilize. So, what does this all mean? We've seen how a journey of millions relies entirely on microscopic glycan shields, and how unseen oxidative villains can tear those shields apart. But as we get better at reading these exact biomarkers, we're moving closer to a world where unexplained infertility is a thing of the past. So, how will personalized biomarker-driven therapies completely redefine the future of reproductive medicine for millions of couples? It's a question that brings immense hope. Thanks for joining me on this explainer, and I really hope this deep dive gave you a powerful new perspective on the microscopic science of life.